Here we go. So today we're doing something that's a little bit different to our previous couple of lectures. We're going to look again at dorsal ventral patterning in Drosophila. So what are we going to do today in this lecture? We're going to look at axis specification in Drosophila. We're going to look at how the dorsal ventral axis specifically is set up in Drosophila. And we're going to look at links between the dorsal ventral axis and immunity in higher animals. So first of all, I'm going to ask you a question, and I know we can't do any of the clickers and stuff, but just have a think of what the answers might be. So when does axis specification occur in Drosophila? And I'll give you 30 seconds or so to think about this. Gave it away. So I apologise for giving it away, but these are the answers that your year group last year gave, and actually, to a degree, all of these are correct, apart from fertilisation. So typically, axis specification occurs in Drosophila from the end of egg formation, that's oogenesis, and it gets strengthened through mid-blastular transition and established finally at that gastrulation stage. So just remember that early Drosophila embryos are just one large polynucleate cell, and we've looked at this before, this concept of a syncytial blastoderm, and for the first 14 cell divisions, a Drosophila embryo doesn't actually have any cell membranes. It has separation between cells, nuclei, so the nuclei are spaced. We can see that here. All the dots in this embryo are nuclei, and for the first 14 cell divisions that remains the case. And Drosophila axis determination starts with this symmetrical deposition of maternal factors. So we've seen this image before. We've seen the bicoid. Oh shit! Sorry. Did I share the screen? I didn't share the screen, did I? So sorry about that. Let's try this again. We won't talk about the axis specification bit. We talked about that. Here we go. Okay, so axis specification occurs in Drosophila when at the beginning and during oogenesis, and then at mid-blastular transition and gastrulation, these axis specification mechanisms are reinforced. And so we have our polynucleate Drosophila embryo, our syncytial blastoderm, and in this all the dots are nuclei, and you get these syncytial blastoderms for the first 14 cell divisions. So what's really important to recognize is that these during oogenesis there is asymmetric, that is non-equal deposition of some of the maternal factors. And here, for instance, we have bicoid, which is whose RNA is deposited at the anterior end of the oocyte before fertilization, and then protein is transcribed, the bicoid protein, the bicoid transcription factor, then diffuses across the length of the embryo, providing us with anterior-posterior specification because of this gradient of bicoid expression. And the same thing happens with Drosophila dorsal ventral axis determination. Again, we have this asymmetric deposition of maternal factors. We have localization of an mRNA called Gurken, and this is deposited between the oocyte nucleus and the dorsal anterior cell membrane. And we can see this here. The Gurken mRNA is detected by in situ hybridization in panels A, B and C. And we can see if we cut cross sections through this where the RNA is deposited between the oocyte nucleus and the dorsal anterior cell membrane. So this region here, we can see dorsal and this anterior, this end is the anterior end of the developing oocyte. And it's this gene Gurken that eventually signals to that protein we've mentioned previously in lectures called dorsal, and this induces ventral fate. So the key thing to remember here is that not only is the anterior posterior axis determined by this asymmetrical deposition of maternal factors, also we get asymmetric deposition of factors which establish the dorsal ventral axis as well. So this mechanism is the last important signaling pathway that we will look at in this course. It's another highly conserved pathway in all animals, and it's used again and again during development, homeostasis and injury. So just a little bit about how this works. So we have our Gurken protein in yellow, and we have an oocyte nucleus that begins centrally in the follicle, but then moves to the dorsal region of the follicle, and this is where the Gurken RNA has been deposited. So because there's a nucleus there, that's where the Gurken protein is expressed, and this is where Gurken interacts with that nucleus. So here in this immunofluorescence image, we can show that the Gurken protein is much closer associated with those cells surrounding the developing oocyte. So this just reinforces this fact that the axis specification dorsal ventral occurs during the process of oogenesis, or egg making. And for our anterior posterior axis, just so you can get a feeling, bicoid is deposited, the RNA is deposited at the border of the nurse cells with the developing oocyte. So bicoid RNA is deposited into the developing oocyte by these nurse cells. So bicoid is produced by these nurse cells, and it then gets deposited into the developing oocyte. So at this end of egg development, in the final oocyte, two axes are already established. So during oogenesis, we can see here that Gurken RNA again deposited in that dorsal region, dorsal anterior region of the developing oocyte. Here, as I alluded to in the previous slide, you can see the nurse cells producing bicoid mRNA, but depositing it in the anterior portion of the developing oocyte. And how this turns out in the end is we have tightly localized RNAs of Gurken and bicoid, Gurken being deposited dorsally and bicoid being deposited anteriorly in that final oocyte. So at the end of egg development, two axes are established. So what does Gurken do? So just so you get an idea, you've probably seen cucumbers. Gurken is cucumber in German. So this might tell you a little bit about what the fly embryo might look like if we have a Gurken mutant. And so a normal Drosophila egg has two dorsal breathing tubes, and you can see these tubes here and here. Our Gurken mutant lacks these because the egg is ventralized. Remember, like the belly piece in Xenopus, this is the equivalent. Gurken ventralizes our egg, and the Gurken mutant has no Gurken mRNA, so because it can't determine dorsal ventral fate, the egg and hence the embryo becomes ventralized. So we can see here that we don't have those dorsal breathing tubes. So this tells us that Gurken is necessary for dorsal fates. So the Gurken protein is a growth factor. Gurken is the homologue of the epidermal growth factor. So another growth factor kinase pathway that we're involved in here. Gurken binds to its receptor torpedo to activate dorsal fate. So what happens step by step is that Gurken is deposited in that dorsal part of the developing oocyte, and then at fertilization the Gurken protein stays there until mid-blassular transition, when we get this cellularization of the oh sorry, my mistake. These are the cells of the developing egg, and the oocyte makes the Gurken protein, and this is received by torpedo. And then it's the activation of torpedo that causes dorsal morphology, and this inhibits the synthesis of a protein called pipe. Because Gurken doesn't diffuse from where it's deposited, the ventral follicle cells make pipe, and the dorsal cells don't make pipe, and therefore this is the region where that breathing pipe is made. So this is a typical growth factor signaling pathway. But just remember, Gurken binds to torpedo to activate dorsal fate. So Gurken is anchored to the oocyte nucleus. This is why that localization of signaling occurs. And so we have our oocyte nucleus with Gurken. Gurken binds the EGF receptor, which is torpedo, and then this signals to the nucleus of our follicle cell during oogenesis. So the next important protein, I apologize, these were all discovered by Germans, so they have very strange names. So the next one is a gene called Spatzle, and this is a soluble extracellular factor in the Drosophila germplasm, which is the contents of the syncytial embryo, and this could induce ventral structures, so extradentical bands. So here's our uninjected embryo. If we took this soluble extracellular factor, Spatzle, and injected it into the embryo, we got more ventral structures, extradentical bands, and more ventral structures. So the purified protein in this factor is Spatzle. So what is Spatzle? Again, it's another food. It's a pasta-like noodle dish from Germany that's kind of twisty. And we can see here in our Spatzle mutants that we get these weird, highly distorted embryos. And this is due to the embryo being completely dorsalized. So in Drosophila, the ventral part of the embryo has all the important tissues in it. So a dorsalized Drosophila embryo looks like a belly piece. So what is Spatzle? Spatzle is another extracellular receptor ligand that binds to another receptor called Toll. This interaction causes the expression of dorsal in the ventral part of the embryo. And you probably remember this from our gastrulation lecture. So this protein, so here's a question, which protein is expressed where gastrulation occurs in Drosophila? So I'll give you a few seconds to think about that. Okay, so you may remember, and this is last year's answers, that there was this protein called twist that seemed to be expressed where gastrulation occurs in Drosophila. So we can see this here. We see twist being expressed, and this is, we're using an antibody to this twist transcription factor, and wherever we see expression of twist, this is the area specifically that will undergo gastrulation, and we can see this here. These cells invaginate and involute, and then only these cells form these mesodermal structures. So twist is a transcription factor that specifies the ventral part of the embryo. This is where gastrulation starts in Drosophila. And like in Xenopus, the tissue that gastrulates becomes the mesoderm. So wherever gastrulation starts, that tissue becomes the mesoderm. Remember, Chordin expression indicates the site where gastrulation begins in Xenopus, and Chordin tissue, Chordin positive tissue, becomes the chorda mesoderm. So very analogous things happening in Drosophila versus Xenopus, except it's flipped on its head, so to speak. It's ventral as opposed to dorsal. So here is our concept of dorsal ventral patterning in Drosophila. Again, it all relies on concentration gradients. So very like we have a concentration gradient of bicoid, we also have a concentration gradient of this gene dorsal. And remember, dorsal will be in a high concentration in the ventral part of the embryo because genes in Drosophila are always named after their mutation effect. So if you have no dorsal, you'll have a ventralized embryo. sorry, you have no dorsal, you have a dorsalized embryo, my apologies, and that's because dorsal is expressed in the ventral part of the embryo. And our concentration of dorsal determines the fate of the tissue during gastrulation. So in high levels of dorsal concentration we get mesoderm. In intermediate levels we get neuroectoderm, and then at low levels of dorsal expression we get the dorsal region of the Drosophila embryo. And here is a cross section with an antibody to dorsal, and we can see that it's expressed in the nuclei of the ventral part of the embryo, and then reduces in concentration towards the dorsal part of the embryo. So again, we have a transcription factor that adopts a gradient during the syncytial embryo stage of Drosophila embryogenesis. So the organizer in Xenopus secretes inhibitors of this protein family. Have a little think about this and then I'll tell you the answer. So almost everybody said BMPs. So typically genes like Chordin are bind and inhibit BMP signaling. I did make a minor mistake here. Hedgehog inhibiting by noggin also occurs in the organizer region and other regions surrounding that. So that's another one to think about. So the dorsal gradient is a morphogen gradient, and like any other morphogen gradient it has an opposing ventral gradient. And we remember this gene decapentaplegic. Remember that from our imaginal discs lecture, and it's this decapentaplegic that forms the opposing gradient. So here's our ventral side of the embryo, and in our dorsal side of the embryo we get the expression of DPP, this BMP in Drosophila. So why is, and this is the confusing thing, why is decapentaplegic, the BMP homologue, expressed in dorsal tissues? Doesn't it inhibit dorsal fate in Xenopus? So remember, in Xenopus, if you overexpress BMPs, you get more trunk tissues at the expense of head and dorsal tissues. So in Drosophila it seems to be deposited in the reverse manner. And that's because everything is flipped in Drosophila compared to Xenopus. So in Drosophila it's the dorsal area that has high expression of BMP, and this becomes mesoderm and dorsal epidermis, whereas the ventral side becomes our neuroectoderm, etc. Whereas in Xenopus, as we know, it's the ventral side of Xenopus embryo that expresses high levels of BMP4, and this drives differentiation to epidermis, muscle and blood. And where we have high expression of noggin and Chordin, or the equivalent in Drosophila is called SOG, and then we have noggin and Chordin expression, we get the formation of the central nervous system and the notochord. So whereas in Xenopus it's the dorsal tissue that has the head and CNS fates, it's the ventral tissue in Drosophila that has the equivalent fates. So everything is flipped in Drosophila. And just to remind you, SOG is Drosophila chordin. So here's another question, you should be able to answer this. Drosophila is a it's a protostome, which means its mouth is developed first, which is the opposite to us and to xenopers, who are both deuterostomes, and their mouth develops second. So remember that during gastrulation in xenopus, the bastopore becomes the anus and the mouth is developed last. And so we have these two different uses of the same signaling pathway in these protostomes versus deuterostomes. So in our idealized arthropod, we have a dorsally patterned circulatory system, and our nervous system and spinal cord are ventral tissues. And this is because decapentaplegic BMP is deposited dorsally, and its inhibitor SOG is deposited ventrally, and this causes high levels of decapentaplegic dorsally, and therefore that is the equivalent of our idealized vertebrate that has high levels of BMP4 ventrally, and in the organizer we get expression of chordin that inhibits BMP4. So in this case we get a dorsal CNS and a ventral circulatory system. So just to show you that these things get flipped in the vertebrate compared to the arthropod. And nuclear dorsal levels can determine the degree of ventral fate. So here we have a wild type and two mutant embryos that can express dorsal over larger areas of the blastoderm. And here in our wild type, snail is like twist, it specifies the area that's going to gastrulate. And in wild type we can see there's only high dorsal in the ventral part of the embryo, and we see localized ventral expression of snail. Once we start increasing the levels of dorsal, and here they reach to over halfway around the embryo, we can see that this closely correlates to the expression of snail, and so to be a much larger domain that gastrulates. Finally, if we express dorsal throughout the embryo, then we get our snail expression or our twist expression throughout the embryo, and then the whole of it will try to gastrulate, which obviously isn't possible. So just to reiterate, nuclear dorsal levels determine the degree of ventral fate. And dorsal can move from the cytoplasm to the nucleus, and it's Cactus that is the protein that controls this. So here we have wild type nuclear dorsal, and in the other cells there is dorsal around, but it's confined to outside of the nucleus. In our toll mutant we see no nuclear dorsal, so the toll mutant prevents nuclear expression of dorsal, whereas our Cactus mutant, without Cactus, this gene Cactus, we get nuclear expression of dorsal throughout the developing embryo. So what happens in a toll cactus double mutant? Remember, the toll mutant prevents nuclear dorsal, and the cactus mutant allows dorsal to always enter the nucleus. So what happens in a toll cactus double mutant? Well, remember that cactus is, you need to know that cactus is downstream of toll. So we know about dorsal, this transcription factor that enters the nucleus. Now, dorsal can only enter the nucleus if cactus is degraded. So if cactus is degraded by ubiquitination, then dorsal can enter the nucleus. Toll is our upstream receptor, so cactus degradation allows dorsal into the nucleus, and it will do this even without the toll mutant being present, because cactus is downstream of toll in this pathway. So one really weird thing that happens when you activate mutations in dorsal, toll and Spatzle, which are our ventralising mutations, a very unexpected extra thing occurred. So what happened was antimicrobial proteins that are expressed by Drosophila were upregulated in a developing larva. And this is this is a Western blot, so we're detecting RNA from our ventralising mutants versus a control. And these 6 genes are our antimicrobial peptide RNAs, and we can see here that in every case of our ventralising mutations we see increase in expression of our antimicrobial peptides RNAs, and this is compared to our loading control, which seems to be a ribosomal protein. So when we activate this pathway, this toll dorsal pathway, we get increased antimicrobial response. So some kind of immune response due to the activation of these genes. And this was further confirmed later on in this paper. You can knock out the toll gene in the adult fly, and when we knock out the toll gene in the adult fly, so we're looking at these black squares, we can see here that very, very quickly we see that flies die. And this happens if you infect them with A. fumigatus or Escherichia coli. Or actually not Escherichia coli, my apologies. So this is specific for this A. fumigatus. And if we look at the fly, you can see they're highly infected and infested. So when toll is knocked out, when toll is knocked out in the adult fly, they rapidly die from fungal and bacterial infections. And in these infections, so when you infect an adult fly, these key components of the dorsal toll pathway are expressed in these fly infections. So here again we have a Western blot for, a northern blot. Why am I saying Western blot? It's a northern blot because we're looking at RNA. My apologies. Northern blot is RNA, southern blot is DNA, Western blot is RNA. And this is definitely a northern blot, so RNA. And here is another northern blot, and we can see here the expression of Cactus, toll and Spatzle message after 3 hours of infection with a fungus or a bacteria. So this same dorsal ventral patterning pathway is also activated in the presence of infection. And so this is a conserved pathway for regulating nuclear transport of transcription factors in Drosophila and mammals. So in Drosophila we have Spatzle, which binds toll, and then through tube and pearl and downstream kinases we inhibit and degrade Cactus, and this allows dorsal to enter the nucleus and initiate a ventralized fate. In mammals, the analogous pathway is the interleukin-1 pathway. Interleukin-1 binds the interleukin-1 receptor, and the interleukin-1 receptor binds MyD88 and through the IRAC and TRAF6 kinases. TRAF6 degrades I-kappa beta, and I-kappa beta degradation allows NF-kappa beta to enter the nucleus and elicit an innate immune response. So we just talked about innate immunity. So this pathway is a link between a classical developmental pathway and a completely unrelated immune pathway in both Drosophila and mammals. So that's the end of this lecture. What did we find out? We found out that not only the anterior posterior axis but also the dorsal ventral axis is specified by deposition of the Gurken RNA in the dorsal region of the oocyte. And then we also looked at how dorsal and ventral fates in Drosophila are flipped compared to those in Xenopus, and we could tell this by where the expression of decapentaplegic was in Drosophila embryos compared to BMP in Xenopus. We looked at that protein dorsal, which when in the nucleus determines ventral fate, and we looked at the fact that these flies, when they are mutant for toll, which is downstream of dorsal, upstream of dorsal, my apologies, then we also get bacterial infections. And so there's this important link between the dorsal ventral axis and innate immunity in Drosophila, and it's that innate immune pathway that's conserved in mammals like us. So I'm going to stop there. I'll end the slideshow and stop share. Right, I think that's it for this time. Why don't we meet back in 15 minutes, so that will be 5 before the hour. Okay, I'll see you then.